Myself, Dr. R.R. Sanap, Head and Associate Professor, Department of Botany, Rao Bahadur Narayan Rao Boronki College, Srampur. Today, I am going to deliver the lecture on the study of Genetum, and in that, the part that is the TS of leaf of Genetum. It is the part of the syllabus of T.Y.B.Sc. Botany, that is paper 4th BO 334, and title of the paper is the Spermatophyta and Paleobotany. So this Spermatophyta, it consists of the gymnosperms and the angiosperms, and Paleobotany. So in gymnosperms, there is one life cycle, that is the life cycle of the Genetum. Today we will see about this transverse section or the vertical vs of the leaf of Genetum. So if we see the general appearance of the leaf of the Genetum, so this leaf, it is the dicot-like. If we see the leaf of the Genetum, it is dicot-like. It shows the presence of the veins, main veins, veinlets having the entire margin and acute apex, and the leaves are petiolate. So these leaves, they are dorsi-ventrally flattened as like the dicot leaf. And if we take the views of the leaf, then it shows the structure like the dicot plant, having the epidermis, mesophyll tissue, and the vascular tissue. So if we see the outline of the section and internal structure of this leaf of the Genetum, then it shows the presence of the outermost layer. So this outermost layer, it is the epidermis. So if we see this epidermis, the cells are somewhat barrel shaped. They are arranged, they are attached to one another, and this epidermis, it is single layered. So thus it is the epidermis, which is single layered. And this epidermis, it is covered by a thick layer of cuticle. So this cuticle, it is nothing but a cutin substance, waxy cutin substance. It gets deposited on the outer wall of the epidermis of the leaf. So thus this is outermost layer, that is the cuticle, which is very important for the regulating the rate of transpiration. So this is the cuticle, which is waxy cutin substance deposited on the outer wall. Then inner side to this, there is the presence of the mesophyll tissue, and this mesophyll tissue, it is differentiated into the palisade and spongy tissue. Palisade and the spongy tissue. So thus here mesophyll, it is differentiated into these two types of the tissues. So the first, it is the palisade. So the cells of this palisade, they are radially elongated or columnar in shape and compactly arranged. So thus here it shows the presence of this columnar cells. So these cells, they show the presence of the chloroplast. So the chloroplasts are present in the cells. So thus this tissue, it is known as the palisade tissue. called as the palisade, which is having the chloroplast. So here it shows the presence of the chloroplast. So as it shows the presence of the chloroplast, so that tissue, it is green colored and it is doing the function of the photosynthesis. And here the spongy tissue, here the cells, some cells, these are the transversely arranged cells, they are transversely arranged, and the tissue which is present in the middle region or the midrib region, here these cells, they are compactly arranged. Cells are polygonal in shape and these are compactly arranged. So it is known as the spongy parenchyma. So here cells, these are compactly arranged without intercellular spaces. So here it is spongy tissue or called as the spongy parenchyma, and this region, it shows the presence of some sclerites in this region. So these cells, these are the transfusion tissue. These are having the sclerite cells. So thus here, this palisade and spongy togetherly, it is known as the mesophyll tissue. So this is the mesophyll tissue which is differentiated into the palisade and the spongy. Then it shows the presence of inner side to it, the vascular bundles. So thus here these vascular bundles, so these vascular bundles, they show the presence of the bundle sheath. So here these are the vascular bundles. These vascular bundles, as I told you, it shows the presence of the bundle sheath. Sheath means covering. So each this vascular bundle, it is having the bundle sheath. And these vascular bundles, they are arranged into the C-shaped manner, and this here, this is the vascular bundle. This is the bundle sheath. And this vascular bundle, it is, if you see this vascular bundle, it is conjoined, collateral, and the xylem, it is the endarched type. So if you see this vascular bundle, it is the conjoined type. So this vascular bundle, it shows the presence of the xylem and phloem. So out of this xylem tissue, it is facing towards the upper side. So thus here, this is the xylem tissue. So the xylem, it shows the presence of the tracheids, vessels, and the xylem parenchyma. and it shows the presence of, below it there is the presence of this phloem. So thus here this is the phloem, which is shown by the yellow color. So thus here this is the xylem and this is the phloem. So here this phloem, it shows the presence of the shoe tubes, companion cells and the phloem parenchyma. So thus here this phloem, it consists of the shoe tubes, companion cells and the phloem parenchyma. Now here this this vascular bundles, they are embedded in the tissue of the parenchyma, that is the spongy tissue. And below it there is a presence of the lower epidermis. So the lower epidermis is present. So as the lip, it is the dorsiventrally flattened, it shows the presence of the upper and lower epidermis. So here this is the lower epidermis, and this lower epidermis, it is also the single layered. But here the important thing is that this lower epidermis, it get interrupted by the presence of the stomata. So here the stomata are present, and the stomata, they are present deep into the region. So here these are the stomata. So there is the presence of the stomatal chamber. Here it also shows the presence of the opening, and here there is a presence of this stomata. So thus here these are the stomata, small pores present on the lower epidermis. So this is the lower epidermis. So actually these stomata, they are embedded deep into the tissue, and through this stomata the exchange of gases and the transpiration is taking place. So thus here if we see this interstructure of the leaf of Genetum, it shows the interstructure like this, having the different types of the tissue layers, that is the epidermis covered by thick layer of cuticle. Epidermis is single layered. Below it there is the presence of the mesophyll tissue, which is differentiated into the palisade and spongy tissue. This palisade, it is the having the columnar cells. Cells are elongated, they are having the chloroplast, and so this palisade tissue, it is doing the function of photosynthesis, and below it there is the presence of the spongy tissue. Some cells here, they are somewhat elongated, known as the transmission tissue, and there is the presence of this spongy parenchyma. So thus palisade and spongy tissue together, it is known as the mesophyll tissue, and inner side to it the vascular bundles are present. These vascular bundles, they are conjoined, collateral, and there is a presence of the endarched xylem. So the protoxylem, it is facing towards the inner side, so it is the endarched type. Each vascular bundle, it is having the bundle sheath or bundle covering, and lower epidermis, it is also single layer. Cells are somewhat arranged, attached one to each other, that is they are somewhat barrel shaped, and this epidermis, it get interrupted by the presence of the small pores called as the stomatal pore, and inner side to it there is the presence of the stomata. The stomata, they are sunken present deep into the tissue. So thus here this is about the internal structure of the leaf of Genetum. Now I am going to conclude here. Thank you very much.